Hi, I am Mrs. Sloan, and this video is all about regulation of gene expression, and this is part 2. In part 1, I went through control of gene expression in prokaryotes, and this is going to be all about eukaryotes. So let me make myself a little bit smaller and get in presentation mode. There I am. Okay. And I need a pointer. Okay, so this is all about eukaryotic regulation of gene expression. So a quick review on this is, remember, we have transcription, whereby you use the DNA and you use that as a template to form mRNA. That is transcription. And then translation is when you use that mRNA in order to build your proteins. And just a little refresher on that, every 3 bases on the mRNA is called a codon, and that codon will be complementary to an anticodon on the tRNA, which will carry an amino acid. A series of amino acids put together will be a protein. And most cells have the same complement of DNA, the exact same DNA in them. And so how is it that our cells are so varied, so different in their functionality? And that all has to do with differential gene expression. So different genes are turned on at different times. And so what we want to talk about today is, how does that occur? So my students have their notes. The notes are also in the descriptor of this video below. And those notes also have a link to this presentation that I'm doing right now. If that would be helpful for you, column 1, please fill in the notes, the scaffolding that I'm providing. In column 2, I encourage you to throw in pictures that are helpful for your understanding and reviewing. So on your introduction, on the notes, I am at 13.2, eukaryotic regulation. And as far as the College Board goes, this would be 6.5 and 6 as far as the unit guides. So introduction, most cells have a complete complement of DNA, all the same genes. Different genes are turned on in different cells, and that's called differential gene expression. So when we look at eukaryotic control, okay, I'm trying to move myself and having trouble. Okay, so when we look at levels of control in a eukaryotic cell, there are 5 main levels of control. The first 3 are all within the nucleus. The last 2 are going to be out in the cytoplasm. So we're going to talk about chromatin structure, and this is referring to just the DNA specifically and its availability for transcription. Transcriptional control has to do with the ability of RNA polymerase to bind to the promoter and make an mRNA copy of the DNA template strand. Post-transcriptional control, post coming after transcription, is any modification to that mRNA after the initial mRNA is made to form a mature RNA, including the cap and the tail and cutting out introns and leaving exons. And also your ability to leave the nucleus. Translational control has anything to do with the mRNA being able to get on the ribosome and be able to translate the mRNA into a series of amino acids. And then post-translational control has to do with folding. Remember, primary, secondary, tertiary, quaternary structure of proteins. Primary structure is the sequence of amino acids. Secondary structure is alpha helix, beta pleated sheet, and that is not dependent on the R groups. The tertiary structure is dependent on the 20 different R groups, right? And their interactions with each other. That folding pattern, chaperone proteins will do that. And then quaternary structures if you have more than one chain. All right, so that's an overview of what we're going to talk about. And then we will finish up our discussion today talking about mutations. What happens if the DNA is screwed up? All right, so we are going to start with the chromatin structure, level 1, and this has to do with just about the DNA. So remember, your DNA is wound. Remember, you have histone and non-histone proteins. Histone proteins are pretty generic, right? And not a lot of variety in those. 8 of them come together to form a nucleosome, and the DNA is wound on the nucleosome. So the more tightly it is wound on the DNA, on that nucleosome, the less accessible it is for transcription. So what controls whether or not it is wound found on those nucleosomes? Well, that comes down to a couple of different things. One is DNA methylation. When it's methylated, it tends to be very tightly coiled around when you have a methyl group on it. Here's one right here, and I'll show you this in a little bit too. And then an acetyl group is a histone modification on this tail right here. And if you have that on the tail, it tends to unwind and make it available. So it's whether it's methylated or not, or whether you have you have an acetyl group on it or not. So we are underneath. Oh, I need to get you caught up. One more thing here, backing up just a little bit. Number 3, you have everything about on the introduction. We have 5 main levels of control. But in number 4, in general, eukaryotic cells don't have operons. Remember operons, and we were looking at that with the prokaryotic cell. Instead, for eukaryotic cells, the genes, you can still control them together because they can be, so you want to fill that in your notes. But genes coding for the enzyme of a metabolic pathway can still be expressed together, even if scattered amongst the chromosomes, because they all share the same control elements. They share the same control elements. Okay, so then the next box down is about chromatin, DNA, and associated proteins. So remember, DNA is wound on histone proteins. The more tightly wound, the less accessible it is for transcription. And this is governed by 2 chemical reactions or interactions, DNA methylation. The addition of methyl groups to the DNA causes it to be more tightly packed and inaccessible. And remember what that is when it's tightly wound, that's heterochromatin. And then histone acetylation, acetyl groups are added to amino acids of histone proteins, thus making the chromatin less tightly packed and encouraging transcription, and that would be euchromatin. All right, so let's look. Remember, I want to kind of review this outer line right here. Let me get a pointer. This outer line here, that would be the nuclear envelope. This is the nucleolus inside. The dark staining material here, that is heterochromatin, that would be tightly packed. So you're going to assume methylation took place there. The lighter colored DNA, the euchromatin, the non-staining, that would be acetylation. It's more available for transcription. So you can see that in this diagram right here, acetylation, regions with a lot of transcriptional activity. And so I think acetylation, and I think active acetylation, that helps me. And so it's more exposed, but look how it's tightly compacted here when it's been methylated. Okay, and that would be heterochromatin. On your notes, I think I gave you all of that. Examples of heterochromatin, so let's talk about that. That would be bar bodies would be a good example. In females, we have 2 X chromosomes, and only one of those X chromosomes is active. The other one is as heterochromatin, and we call that specifically that heterochromatin of that X chromosome, we call that a bar body. And you can see a bar body inside this nucleus right here. So it's the second X condensed, and females are considered mosaic because you never know which X is going to be condensed in which cell. And you can see that in calico cats due to which X chromosome is getting expressed. Also, you can see that sometimes in different colored eyes. So you could have twins that have different colored eyes depending on what genes they've inherited in their X chromosomes, because they could have the exact same X chromosomes, right? If they're identical twins, and these would be just girls, right? Because they're the ones who have 2 copies, identical twin girls, and which X is getting expressed in those twins can affect what their eye color is. So on your notes, I think it's all filled in for you. Yes. Okay, and then lamp brush chromosomes. This is an example of euchromatin, and this is where parts of the DNA is looped out. It's called lamp brush because those would be kind of old school for cleaning lamps. Think of it like a test tube brush and how you have those bristles coming out there. So that's showing you what DNA is available and would be considered euchromatin. And so I gave you everything for that too. Epigenetic inheritance. Epi means above, so above the gene. So think about that, right? You could have what you inherited, but if that gene can never be expressed, right, epigenetic inheritance, because of its methylation, then you won't have that particular trait. So on your notes on epigenetic inheritance, inheritance patterns that do not depend on the genes themselves. They don't depend on the genes. They depend on whether those genes can even be available for transcription. And then genomic imprinting, I gave you that when DNA is methylated during gamete formation, which then ensures passing on that information of what groups should be methylated or which regions of your DNA. Okay, that brings us then to number 2, still in the nucleus. This is transcriptional control, and this is the most critical level of control. So on your notes, you want to add that in, most critical level of control. So remember, we're not dealing with operons, what we learned about with prokaryotic cells. It's more about transcription factors helping RNA polymerase just bind to that promoter. So when you look in a very simple Minecraft almost diagram here, so remember when we were looking at how RNA polymerase has to bind to the promoter, you could have repressors that are involved, and then as long as that promoter is available and facilitated by a transcription factor, then it would transcribe those genes. So this is just showing you transcription factor so that you can get that protein. And on your notes, you have transcription factors, proteins that assist RNA polymerase to bind to that promoter. Now, in eukaryotes, there is a conserved sequence. So that means you find it again and again in eukaryotes. And it is part of the promoter sequence called the Tata box that you see over and over again. And it's a series of bases on the DNA where it's just TATAAA. And you see that again and again and again. And so on your notes, you have underneath transcription factors number 1, in eukaryotes, a Tata box is part of the promoter. And then the little letter B should be a conserved DNA sequence, and then I gave you what it typically is. So what would this be evidence for? Evolution, right? When you have that same sequence of DNA over and over again. All right, now let's level up. Besides transcription factors related to that Tata box that I just told you about, there are also transcription activators. And they have their own little area that they bind to, and that's called an enhancer region, and that would be farther upstream. So you could have here, you can see right here is your promoter, and much farther upstream, you could have an enhancer region, and you could have more than one enhancer region. And the transcription factor will not bind onto the promoter unless you have the activators on the enhancer region, and then it forms this hairpin loop that assists the transcription factor to bind on. Do you remember when we talked about cyclic AMP in prokaryotes and how cyclic AMP, the levels are higher when you're low in glucose? And that was to facilitate that you only make the enzymes to digest lactose if you're a bacterium, if you have the presence of lactose and the absence of glucose. So you wouldn't activate CAP, a transcription factor, unless you had that cyclic AMP. Here in eukaryotes, you need to have these activators that bind to enhancers that help the transcription factors to bind and help RNA polymerase to transcribe those genes. And on your notes, let's talk about the transcription activators. You actually have all of that on your notes already, so you don't have to do anything more on that. So this is to show you all the things. You could have several. Here you can see 1, 2, 3, 4 different enhancer regions, all helping RNA polymerase to bind to the promoter. And look, there's that Tata box that we talked about. So the light is green when all of these things are in place. And this is truly just an oversimplification. It can get much more complicated than what I have shown you. And all of these things are to tell you this. For eukaryotes, it's not about negative control. It's really about positive control. You have to have all the right conditions in place before you transcribe those genes, right? And so it's kind of a checks and balances to make sure you only transcribe the genes that are necessary and that won't harm that particular cell or environment, right? Okay, so then, I just threw this in here and I don't know why. There's nothing on your notes about this, but I wanted to remind you about jumping genes that could throw that off. Those are called transposones. And this is when in the DNA, it self-excises and puts itself somewhere else. And where we see evidence of transposones is in Indian corn. So this is where the DNA itself is kind of moving around, and that can definitely influence transcription if it jumps into the wrong place. All right. And then this is our third level and our last one within the nucleus. This is post-transcriptional control. So this would be the DNA has been available. RNA polymerase has been able to get on the promoter, and I made some mRNA. Now, what am I going to do with that mRNA once it's made? You know what we learned about before, the cap and the tail and cutting out the introns, right? So post-transcriptional control, okay, first, look at the very top. You have your DNA strand, and then you have your green mRNA right here. I want to point out to you, just as a review for AP, that RNA could go out and be tRNA, right, which will pick up amino acids. That RNA could go out and be ribosomal RNA and be part of a ribosome. Or it could be one that's actually going to be translated, right? So this pre-mRNA right here, we're going to snip out this intron, right? Remember with SNRPs? And once you cut it out and splice the exons together and you've got a cap of 7-methylguanosine and your poly A tail, maybe 100 to 200 adenines, that would be a post-transcriptional modification. And also post-transcriptional control is, does it have the ability to leave the nucleus itself and to get out there? That is also considered post-transcriptional control. So on your notes, you have everything for post-transcriptional control, starting all of number 1. But number 2, speed of transport of mRNA through the nuclear pores, All right, then on top of that, let me show you another picture on that, just showing you exon recombination. So here you have your genes, A, B, C, D, E. Here's your mRNA, and you have cut out the exons. And and as a result of this mature mRNA with its cap and its tail, you make protein product 1. But notice, what's the difference between 1 and 2? A, B, they removed C. They treated C as an intron and removed it. So it just goes A, B, D, E. So now we have a different series of bases in our mRNA, which will give you a different protein product. All right, so that is exon recombination. Now, other things can get in the way, and I know this is a scary chart, but I just wanted to show you because there are so many different things here that can happen, and this affects not only, if you look up here at the top, RNAs involved in post-transcriptional modification. It could interfere with DNA replication, and I'm going to show you one that could also affect translation as well. It could interfere in any one of those areas. And that's because, remember, DNA is double-stranded, but RNA is single-stranded. And so what can happen is anything that can bind to that RNA and making it double-stranded with complementary bases, then it can influence how it is processed, modified, whether it can exit the nucleus, whether it can eventually be translated. So on your notes, I have small RNA, sRNA, can regulate transcription and translation. And it could be originated from double-stranded DNA, which we've talked about before. So transcribed RNA loops can become double-stranded RNA. Double-stranded RNA is cut up. The double-stranded RNA is cut up to form small RNAs. What will those small RNAs do? They can regulate, that's the word you need to put in there, both transcription and translation. And they are a source also of microRNAs. Now, look at all the different kinds of RNAs that you can have, small nuclear RNA, and what their function is, right? Now, look, it's not like it's all bad. Look, tRNA maturation. So I don't want you to think of these as bad things, just regulatory. Can some be bad? Yes. All right. Here are some regulatory RNAs. So short hairpin RNA. Look at all the different ones in here. One's related to CRISPR, right? Remember we talked about, we will talk about CRISPR. So on your notes, microRNAs can bind to mRNA inhibiting translation. Small interfering RNAs form a complex with an enzyme called RISC. RNA-induced silencing complex, and it degrades any mRNA the complex is complementary to. Right, so there's a lot of different options there. We don't need to go into the details of it other than you know that it can be regulatory in function. Okay, and then translational control. Translational control, remember, this is what's going to occur out in the cytoplasm. These last two occur in the cytoplasm. And basically, anything that can impact the ability of that mRNA to get with the small and large ribosomal subunit and tRNAs to the anticodon to bind with the codon. So translational control has to do with what I just mentioned previously, any of those interfering RNAs, but also how long the mRNA lasts before it gets degraded. Any mRNA modifications, and like I said before, those microRNAs. So underneath translational control, I gave you everything. So there's nothing you need to add to your notes on that one. Okay, and then the last one of eukaryotic regulation, the fifth one, also out in the cytoplasm, is post-translational control. So at this point, you have major protein, but remember we talked about you've got to get it in the right shape. So post-translational control, proteins are continuously being formed and degraded. You only keep what is needed. You only keep what is needed. So it might have to have additional changes. It has to be folded in a certain way, and then it has to last, right, in order to be functional. And I gave you that misfolded proteins can cause problems or aggregate. Things like Parkinson's disease and Alzheimer's disease are related to that. And lysosomes can help degrade those unless you have issue with your lysosomes. All right, so those were the 5 big levels of eukaryotic control of gene expression. And then this is just a little bit of an overview I wanted to show you. So you have transcriptional control, post-transcriptional control, you have translational control, and then post-translational control would be the folding pattern. And then this would be talking about chromatin, whether it's even available or not, whether it's euchromatin or heterochromatin. All right, the last on this topic is when your DNA is bad, then obviously then your proteins can potentially be bad, and those are gene mutations. So on your notes, mutations are the primary source, if it works out well, of genetic variation, and it's defined as a permanent change in the sequence of bases in DNA, a permanent change in the sequence of bases in DNA, and its impact could be nothing to complete inactivity. Nothing to complete inactivity. So let's take a look at some different options here on genetic mutation. So here is the codon as it should be. So here in your DNA, ATG. That's what you're hoping for. The mRNA, remember, complementary, A with U, T with A, G with C. And if you look up this codon, it codes for tyrosine, and that would be part of an amino acid, which could give you a normal protein, Now, what I'm talking about right here are what's called point mutations. So this, you can point to a single nucleotide that has an error in it. So in this case, your DNA is ATA. So when you make your codon, instead of being UAC, it's UAU. Well, remember how we talked about how you can have more than one way for several or most of the amino acids, more than one way to get the exact same amino acid, right? There are multiple codons. So you will still get tyrosine. And this is what's called a silent mutation because it's wrong, but it still gave you the right amino acid, so you still have your normal protein. But this point mutation right here, when it mutates instead of ATG, it's ATC, then you have UAG. This is one of your stop codons. So you will have, this is called a nonsense mutation because you could be right in the middle of a sentence, right? And it stops. So it interferes with that sequence of amino acid. I was trying to model that for you. And then this last one, the very first, which is always the most dangerous, the very first one in the codon mutated. So you get histidine, and this is a missense, and this would be a reason why you would have sickle cell disease. It's a missense mutation. It coded for the wrong amino acid. So just silent, no effect. Right? Nonsense, stop the protein mid-protein. And this one you get the wrong amino acid and you get the wrong probably folding to it because it is not the same amino acid as before. So on your notes for that, on the impact, if it's in germ cells or gametes, it's definitely going to get passed on because it'll be then a member of every single cell in that developing zygote. If it's a somatic cell, it impacts a small number of cells, but then it could lead to cancer. So what causes those mutations? So it could be spontaneous, associated with any normal process gone wrong, or remember what I talked to you about, those transposons, those jumping DNA could also cause that. It could be a result of being exposed to some sort of toxic chemical as well. So on your notes, I'm going to go, I kind of put these a little bit out of order. So jump down onto, and I'll come back to that portion on the causes of mutation after I talk to you a little bit more. So jump down into your notes, into the effect of mutations on protein activity. Point mutation involves a change in a single DNA nucleotide, which can alter transcription and possibly change the amino acid it coded for. And I gave you some examples. Now I'm going to give you another type of mutation, and this is called a frameshift mutation. Now, because we don't have the codon chart memorized, instead of doing 3-letter codons with the bases you know of DNA, right, A, T, G, and C, instead I've changed it into 3-letter words, right? Can you read this sentence right here? So hopefully you can. I'm going to space it out now to make it easier. So the big fat cat ate the big rat. And those are modeling for you codons. So a mutation might be where you have a deletion, okay, of the E in this the. So then your first codon is no longer going to be THE, right? It's going to be THB, and it's going to shift all the next codons are all going to be off. So instead of the big fat cat ate the big rat, you have the if a attic tet have a igor at, right? Which makes no sense. And that's called a frameshift mutation. So on your notes, as a result of either an addition or a deletion in this case, but it could be an addition or a deletion, codons are reset from that point forward. And then all of these can form non-functional proteins. So here's an example of that. So phenylalanine, you have an enzyme that converts phenylalanine into tyrosine, a different amino acid. If you don't have that enzyme, you will have PKU, and we've talked about PKU before. Additionally, this tyrosine needs to get converted into melanin, a pigment, right, for coloration. If you don't have that particular enzyme, you'll never convert your tyrosine to melanin, and as a result of that, you will be an albino. So that's just kind of showing you if these proteins aren't active, what are the consequences of that. So here is one called androgen insensitivity. She is an XY. So genetically, she would be a male. And she has testes in her abdomen, but she has faulty receptors. So the DNA that would code for the receptors for the testosterone, remember, think about cell signaling. The testosterone is that signal. The receptors, the receivers of that cell signal, don't have the right shape to bind to that. And so as a result of that, she defaults to being female, Okay. Also, a little review, this can cause cancer is a problem, right, with mutations. And it is estimated, like, of all the children that were born in 1999, and I don't know what year you were born, but of children born in 1999, a third of them will develop some sort of cancer, and a third of those that do probably will die of that, right? And a fourth of the males will die from that. Those are just cancer stats, and I have that in your notes. Mutations can cause cancer. Chances 1 in 3 expected to have some sort of cancer. And then I gave you the rest of that. The 3 deadliest are lung, colon, rectal, and breast. And then remember those genes we learned about when we talked about the cell cycle, right? So proto-oncogenes are in positions of control, and they can, if they mutate and become an oncogene, they are like facilitating the cell cycle even though they haven't received the signal. And if you remember, we talked about the RAS protein. RAS is like stepping on the gas. And so even though it hasn't received, let's say, a growth factor to trigger the RAS, it still goes and that gene expression takes place. And so that promotes the cell cycle. Okay, so that was one that we talked about. And then the other gene that you would be concerned about mutating are tumor suppressor genes. And the reason tumor suppressor gene, remember about P53 and caspases, they are the ones that when you have cells out of control, they're to eliminate them. And if they're like your assassins. So if they're not working, then you don't eliminate those cells with that mutated, where a proto-oncogene has become an oncogene. And the one we talked about the most was the P53 gene, and the checkpoint in the cell cycle at G1. So what kind of things can cause cancer? So now jump back in your notes where it says cause of mutations, and I already talked to you about spontaneous and then induced, and it can be induced. It may result from exposure to toxic chemicals. And mutagens are environmental factors that change DNA, and you want to fill that in, that change the DNA. It's called a carcinogen if it causes cancer. Common mutagens are things like radiation, organic chemicals, smoking. These are all mutagens, right, and carcinogens. So there is what's called the Ames test to see if it's mutagenic. And so just showing you right here, they will take a specific strain of salmonella that doesn't have the ability to produce histidine, and they will mix it with a possible mutagen and see after it incubates, does it have the ability to grow, meaning that it's been put on a medium that does not have a supply of histidine. So if it can still grow, it must have gained an ability, so it mutated. And so it could grow on this medium. And they will compare that, if you look down below, to its control right here, because there can just be spontaneous mutations. So you want to eliminate what percentage of it would probably be due to a spontaneous mutation. Okay, so they will check using the Ames test to see how bad of a mutagen it is by how it causes this salmonella to fix itself with a mutation. And so I gave you that already in your notes. And then I also mentioned that DNA repair enzymes, which constantly monitor our DNA, can handle different types of mutations and try to fix those for us. And that, my folks, is the end. And if you are one of my students, I'll see you in class.